Hello and welcome to this National Pathology Week video. My name is Dr. Susie Lishman and I'm President of the Association of Clinical Pathologists. This series of videos on pathology myths and misconceptions was recorded as part of National Pathology Week 2024, an annual celebration of the vital role that pathology plays in everyone's healthcare. It was developed by the Pathology Alliance, an umbrella organisation of all of the UK's pathology societies and organisations. In this video, Emma Holt and Rosemary Felix will explore the statement, "Haematology is all about counting blood cells."
So hi everyone, I'm Rosemary and I'm joined by my colleague Emma today. We're both trainee clinical scientists in cancer genomics based in HMDS in Leeds. Today we're going to try to talk about some of the different methods we use in haematology lab to try and convince you that haematology is more than just counting cells. Next slide, please. So this is probably what you might think is cell counting, where you see clinicians looking down the microscope. This investigation is actually called morphology. And when you break down the term morphology, it essentially means the study of shape and form. And that's exactly why we use this technique. As you can see in the figure, a drop of blood is spread on a glass slide, stained and examined under a microscope. This gives a snapshot of the cells in the blood at that exact moment. Observing cell shape and appearance helps us detect any changes that might indicate disease. So here you can see a normal blood smear, which is a lot of these red blood cells and a few of your white blood cells. So next animation, Penny. But this is an example of how a person's blood cells will look like if they have sickle cell. Next animation. And this shows the cell of a patient who's been infected with malaria. And next animation. And finally, this is an example of what a scientist or clinician in HMDS will see in a patient with chronic myeloid leukemia. Next slide, please. Now, this is the bit where cell counting is actually a thing, but we get a machine to do it for us. So imagine you have a mixture of different cells in a blood sample, and you want to understand more about their type, size, or specific characteristics. Flow cytometry lets us study individual cells by having them labeled with these fluorescent markers that attach to specific features. So when a cell passes through one by one through a laser, detectors capture how the light scatters, revealing the cell size. The fluorescent markers, color brightness, then helps us to count cells, distinguish different white blood cell types, and assess their maturity. So you can see in the figure here, the different cell types are mapped to distinct locations on the graph so that you can see what cells are present and how much of them in proportion to other cells in the blood. So next animation, please. So this figure is showing you the results of a normal sample and a sample of a patient with acute myeloid leukemia. You might notice that the person with leukemia has a much denser population of immature cells called myeloid blasts, which is the thick red section that you see. And these shouldn't really be seen in healthy circulating blood. And so when a clinician looks at this graph, they can see that the patient potentially has leukemia. So essentially, flow cytometry allows us to look at individual cells in ways we can't with our eyes alone, making it an essential technique for modern diagnostics and research. Next slide, please.
Thanks, Rosemary. I'll take over from here. So as well as looking at cells as a whole, we can also look at how the cells are built. To do this, scientists can look at genetic material that forms the blueprint of the cell, the genome, which I like to think of as a book. So our book of the genome is made up of letters which are the DNA bases. Now, it's quite a boring book because DNA is represented by 4 letters, A, C, G, and T. Our DNA words come together to make paragraphs, and in our genome book, each paragraph represents a gene. A collection of paragraphs in a book forms a chapter, and in our genome book, collections of genes make up chromosomes. Humans have 23 pairs of chromosomes, and the summation of all of these chapters in our book makes the genome. And this contains approximately 3 billion DNA bases. Our book with 23 chapters is stored within the nucleus of our cells, which, to put this into perspective, it's about 50 times smaller than a millimeter. And that's a heck of a lot of information to store in such a small space. Next slide, please. Overall, we know that our DNA is written in a specific way in our genome book to make a specific gene. Why is this important? Our genes provide the blueprints for how proteins are made in our body. 3 bases of DNA form what's known as a codon, and this provides the instructions to make amino acids. And these are like the Lego pieces that build proteins. Proteins within our cells have a multitude of functions, from producing energy from the cell, helping cells communicate with one another, giving cells special abilities such as being able to destroy viruses and bacteria. They help cells grow and divide, and they ultimately orchestrate the life and death cycle of our cells. The function of these proteins depends entirely on them being made correctly using the right blueprints. So overall, the correct blueprints make a happy cell. Next, please. A change in a DNA, such as a single base change, deletions or insertions of genetic information can influence a change within the amino acid sequence. A change in the amino acid sequence can cause a change to the protein, causing it, for example, to be lost completely, to change its abilities, to change the number of proteins in the cell, and more. So these changes in proteins can impact the cell as a whole. For example, a protein with an additional ability can cause too many cells to be produced, or a loss of a protein can cause the cells to be stuck in an immature state. Or cells could stop communicating with each other and avoid destruction when they become faulty. So, considering how these DNA changes are so small, how do scientists investigate these changes in the lab? Next slide, please. We do this by sequencing DNA. We can take a blood sample and isolate the DNA from the cells. So here's our patient DNA in the well-known double helix shape. We break apart the helix using chemicals and heat, and then the sequencing process can begin. We use matching bases with a fluorescent tag to add to the DNA, and when the correct base is added, it will join to the DNA strand. As the fluorescent base is added, a laser in the sequencing machine causes the tag to emit fluorescence according to which base is added. So here we found in position 1 an A has been added, which emits a purple light. This process is repeated, and at position 2 we can see a C has been added as the sensor detects blue light. This happens millions of times during sequencing, and at the end of the procedure we can combine the information from all of the calls together, and now we know our sequence. So what does this look like in practice? Next animation, please. Our output from sequencing is a string of letters, as you can see here. Through huge studies of thousands of people, scientists have made a reference genome, which is like a general picture of how the genome should read in a healthy person. Our example patient here, unfortunately, has suspected blood cancer. When we compare our patient's DNA sequence to the reference genome, we can see a specific change in the DNA from a C to a T. Clinical scientists will use tools and scientific evidence to work out the impact of this change on DNA and to determine if it's a feature of the patient's disease. Next slide, please. Zooming out a little, we can also use the microscope to look at whole chromosomes, which each present with a unique pattern when stained. Sometimes errors happen when a cell divides, and this can result in changes to the structure or number of chromosomes and subsequently result in genetic conditions. For example, a well-known one is 3 copies of chromosome 1 that causes Down syndrome. Next slide, please. An example of a common chromosomal rearrangement is when the bottom of chromosome 9 and chromosome 22 are broken apart and rearranged. 22 breaks off and fuses to the bottom of chromosome 9 and vice versa. This fuses 2 genes known as BCR and ABL together, and this fusion gene creates a fusion protein that has additional abilities, making the cells divide rapidly. We can see this fusion gene by a technique known as fluorescence in situ hybridization, which has the quite enjoyable acronym of FISH. Next animation, please. This particular FISH technique uses a red fluorescent tag that attaches to the ABL1 gene and a green fluorescent tag that attaches to BCR. If this fusion is present in cells, the red and green signals come together and make a yellow signal where they overlap, and this can be seen down the microscope. Next slide, please. We've given a brief insight into the extensive field of laboratory hemato-oncology in the time that we have. So as well as finding out a patient's specific genetic or cellular changes, these results can be used to inform the patient's diagnosis according to their genetic status. It can be used to assess the prognosis, that is the risk of the disease, how they might respond to treatment, and to inform the treatment strategy itself. So if the patient is eligible for personalised drugs or clinical trials. Rosemary and I would like to thank you for your attention this evening, and we hope we've given you an insight into the hematology isn't all about counting blood cells.
I hope you enjoyed learning the facts behind that myth. You can see more of these videos on the Royal College of Pathologists YouTube channel @ILovePathology.